Okay, so viral structure, anything else? So, some viruses actually do carry enzymes within themselves in that central core to help with specific viral operations. So, some carry polymerases. Polymerase is used to synthesize both DNA and RNA. Depends on which one you're using. There's DNA polymerase and RNA polymerase. But ultimately, what they do is they help to synthesize the DNA and the RNA. Replicases are used to copy RNA. Reverse transcriptase, remember I said that we had the retroviruses? These are used to make DNA from RNA. Each one of these is kind of specific to the type of virus that it's in. Some actually take souvenirs from their host cell to use for later. So, retroviruses, for example, can borrow the host's transfer RNA molecules. So, again, it just depends on which virus you're talking about. That'll kind of give you a better general idea of what's in it, what it might have within it. Okay, so modes of viral replication. There are five main phases in the life cycle of an animal virus. There's adsorption, you're adding it, penetration or uncoding, synthesis, assembly, and then release. So the length of the viral replication cycle, how long does it take, varies. Poliovirus takes about 8 hours, whereas herpes virus takes about 36. So it really kind of does depend on which one you're talking about. Again, you have to kind of get to the specifics of this virus does that. Okay. This is what we're going to talk about. It's those 5 steps. In a greater amount of detail. So, adsorption. A virus can invade its host cell only through making an exact fit with specific host molecules. So, remember what I said about the spikes. The spikes are like having a key that fits into a specific lock. Depending on the tissue in the body, depending on the animal that it's infecting or plant that it's infecting, if you don't have the lock that fits the key, you don't get in. Okay? So we call this a host range. Think about a range of things. Okay, host range. Um, it's a limited range of cells that the virus can infect. So, for example, hepatitis B, specifically in this example, can only infect liver cells. So, if I were to breathe in hepatitis, if I were to put hepatitis on my arm, wouldn't matter because those cells are not liver cells. They don't have the right lock that fits my key. Okay? Poliovirus is actually intestinal and nerve cells for primates, specifically for primates. So remember, we are primates. Primates are animals that have nails, basically, like ours with the bed of the nail. So you've got chimpanzees, you've got orangutans, you've got gorillas, you've got lemurs. These are all primates. So when it comes to poliovirus, it can infect all of those. That's its host range. Whereas if I were to try and infect a dog, it wouldn't work because, again, uh, the virus has a key and it doesn't fit the locks that are on the cells of a dog. Okay. Rabies. Rabies has a huge host range and it can infect pretty much all the mammals. So, bats, dogs, people, whatever. It has a spike or a key that fits a lot of different locks. Or I shouldn't say a lot of different locks. It has a key that a lot of different species have the exact lock that it fits into. So when it comes to, you know, again, hepatitis B, humans and only the liver. But when it comes to rabies, it's a huge window compared to that small window for hepatitis B. Cells that lack a compatible virus receptor, the thing the spike binds to, are resistant to adsorption and invasion by that virus. So, one of the reasons that we wear masks, okay, for this COVID pandemic is because COVID binds to a specific receptor in our lungs. If you're wearing a mask and it can't get into your breathing tubes to get down to your lungs, you don't get it. Somebody with COVID may, lick your arm and you still wouldn't get it. Now, admitted, you need to wash your arm and make sure that you don't put your hands in your mouth and all of that. But honestly, if somebody who had COVID licked your arm, it wouldn't matter if you didn't inhale that virus. Because again, the only place that you have that compatible receptor for the virus is in your lungs. Now, for a while there, they were worried about the eyes, so people started wearing the visors and stuff. But I heard about it, and then I never heard anything else about it. So I'm guessing that it wasn't necessarily true. Okay, tropisms. Specificity of viruses for certain tissues. Like I said about the flu goes to your lungs. You can put the flu in your leg, you can put it in your ear, you can put it in your eye, and it's not the right tissue for it to infect. Again, we've got that window. And if you're not in the window, you don't get it. Okay? So if you look here, if you've got an enveloped virus, those spike proteins are on the outside of it. So when they fit the receptor, it gets absorbed or adsorbed. Okay? The non-enveloped virus still has spike proteins. They still have to match the receptors on the host cell. Both of these viruses, regardless of whether they are enveloped or they are not enveloped, have to have matching receptors to their spike proteins. If they don't, they won't successfully attach. They can't get adsorbed. Okay? So, step 2, penetration and uncoding. So, there are two ways that this virus can enter a cell or penetrate the cell. One of them is by the process of endocytosis. Endocytosis means that you are swallowing, okay? You're engulfing and swallowing that virus. So the virus is engulfed by the cell and it's brought in through a vesicle. Vesicle is a very fancy way of saying bubble, okay? So if you look here, there's the virus, and we actually are surrounding that virus with the cell's membrane as we bring it in. So we get into the cell, and eventually that vesicle and envelope break open, releasing the genetic material that can now take over the machinery of your cell. The other way that we can do this is by a process called fusion. This happens pretty much with any virus, enveloped or not. Fusion, however, only works with enveloped viruses. You'll see why in a second. So, an enveloped virus, remember it has the host cell membrane? You're actually going to fuse that membrane. So, if you look here, there's my enveloped virus, right? When I get to a certain point, that part of the envelope that was the host cell's membrane actually gets incorporated into the host's membrane, cell membrane. And the only thing that really gets put into the cell is the capsid without the envelope around it. Okay? Once inside, the virus needs to get comfortable, basically take its coat off. The capsid needs to get out of the capsid, expose the nucleic acids, and then it basically gets down to business where it starts to replicate itself. Uncoding is a process of the virion shedding the capsid to liberate the nucleic acid. So here you can see that this is broken and the genetic material is out. This would be the uncoding, okay? So here you can see it as well, where here it's a solid virus, but here there's the genetic material coming out. It's uncoding so that the genetic material can go to take over the machinery to start making copies. Okay, let's see if this will work. Give me a second. Let's see. Doo-doo-doo. I'm going to make the volume on this computer as loud as I can. And if you can't hear it, then I would suggest you download the PowerPoint and play it on your own computer. Okay.
There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
Okay, so we talked about in this slide the endocytosis, which you saw in the second example, where it completely surrounds it and brings the whole thing in. The first example showed you this, where the envelope fuses with the membrane and only the virus capsid unit gets in. Then it showed you one that didn't have anything on its surface, no envelope at all, and it pretty much works the same way as the endocytosis. I'm going to envelop it and then I let it out. Okay? So let's talk synthesis. To synthesize means to make. So this phase is where the virus genome, the genetic material, whether that is DNA or RNA, is replicated. I start making copies. And I start making the protein units. Remember, I said they're all kind of triangles in the example. I start making those protein units for the capsid that I'm going to make. Remember, they're a repeating pattern, but they fit together to make that capsid. So in DNA viruses, they enter the host cell's nucleus. They actually go into the nucleus and they're replicated there. In RNA viruses, I don't have to make it into RNA. It already is RNA. So literally, I can replicate that in the cytoplasm. Retroviruses, go back. They turn their RNA genomes into DNA first and then start replicating. Okay? So we call them retro because it's kind of like you're taking a step back. Okay? So that's step 3. Step 4 is assembly. In this step, you're actually putting together the new viruses. Remember, I said when we're making the units of the capsid, we're making individual units, but you kind of have to fit them together like Legos to make an entire capsid. So I'm putting the parts together. And then as I put them together, I'm making new capsids. And remember, I was making copies of my nucleic acid code, whether it be RNA or DNA. So I need to make sure I put that code into the capsid. So I'm making new virus, okay? All of the proteins for my capsid are made in the cytoplasm and they have to join up with those nucleic acids. So for DNA viruses, the proteins that I'm making my capsid out of have to go into the nucleus first, and then they get assembled with the nucleic acids inside of the nucleus. With RNA viruses, the proteins and the nucleic acids join together in the cytoplasm, not in the nucleus. Remember, for our cells, for human cells, Our DNA is only in one place, and that's in the nucleus. So this makes sense. If I've got a DNA virus, it has to go into the nucleus because it's the only place DNA exists inside of our cell. Okay. Now, step 5, release. The number of viruses released by an infected cell is variable. It's controlled by the size of the virus. If you have a really small virus, you can probably pack a lot of them into one cell. But if you have a really large virus, you probably can't, just because of the size limitation of the cell. The other thing is the health of that host cell. Is it a healthy cell that can mass produce virus well, or is it a sickly cell that will do its best, but it's not really going to work very well? These two things play a role in how many viruses get released. But again, it varies by virus. Okay? Okay. There are two ways to be released or leave the host. One is lysis. Lysis means to break or burst. Okay? The other is called budding. Okay? So, lysis. Basically, you are going to burst the host cell. Usually, this is the preference for non-enveloped viruses. So if I don't have an envelope, they multiply and multiply and multiply and multiply until the cell explodes. Okay? When the cell explodes, virus goes everywhere. The thing about lysis is you kill the host cell. So once it explodes, you're done with that cell. It can't keep making virus. Budding is a bit different. You release virions through the host cell's membrane. This is the stage that the virus gets their envelope. Only enveloped viruses are released this way. So remember, I said that envelope comes from your host cell's membrane. So if you look here, here's my capsid, and here's my genetic material, and here are my spike proteins in the membrane of the cell. I get surrounded by the membrane and basically exocytosed. So I'm not filling up the cell until it bursts. I'm literally taking part of the cell with me, the membrane. The spike proteins I programmed to be there, though, as a virus. So again, I'll let it play, and hopefully you can hear it. If you can't, go into Blackboard, download this specific PowerPoint, and you should be able to watch it on your own computer. Okay.
Enveloped viruses are usually released from the host cell by a budding mechanism. First, viral spike proteins are inserted into the host cell membrane. Next, the inside of the host cell membrane becomes coated with viral matrix protein. The viral capsid then becomes completely enclosed by the region of the cell membrane into which the spikes and matrix protein are embedded, and the virus is released by budding.
So again, I'm taking part of the cell with me. I'm not causing the cell to explode. With the lysis method, it's almost like overinflating a balloon. You're filling it and filling it and filling it until it bursts. Like I said, once it bursts, virus goes everywhere. But that cell that is basically being lysed, it's gone. This type of emergence is actually not necessarily as deadly because I can keep the cell making new membrane and everything else, so I can just keep budding off of it. Okay, so viral damage to the host and cytopathic effects. So remember, cyto is cell, okay? During the course of a viral infection, the virus can make changes to the host cell that are known as cytopathic effects. Cytopathic effects can range from very minor effects to fatal effects. Okay? Depends on the virus, depends on what it does. So, some cytopathic effects, some of the things that you see with certain viruses, there can be changes in the shape or size of the host cells. If you look here, that's a normal cell, and then look at these giant cells, completely different than the normal host cell. You can form inclusion bodies. See these dark spots, these are compacted masses of virus or damaged cellular organelles. We do see a lot of inclusion bodies when somebody has the rabies virus. Okay, it's basically a holding tank, but it's for masses of virus and damaged cell parts. Formation of syncytia. So syncytia are large multinucleate cells, and these are formed by fusing together host cells. So here, if you look down here, you'll notice that I have a lot of nuclei. This is one massive cell that was the fusion of several cells into this one kind of mega cell. These changes, this is not normal for a human body, right? Our cells are individual. They don't fuse together like this. They don't become these ginormous cells, and we normally don't have inclusion bodies. So this can cause damage to the cell. It can cause death to the cell. So these changes aren't the best thing ever for our cells. So, types of viral infections. I think I'm going to stop there for part 2, and I'll come up with part 3.